Good afternoon. How's everybody doing? Good. I'm Keith from GRI, and I'm excited to welcome everybody to GRICon. We'll have a more formal welcome tomorrow when we have our first full-day session with all of the families and researchers together, but excited about this today. So a couple of things I just want to do to start is to thank all of our sponsors. We wouldn't have been able to do this conference without them, so thank you to everyone. And a special thanks to our diamond sponsor, GRI Therapeutics, who's really came up big time for us and allowed us to put on a really great conference. So thank you to all our sponsors. Here's the network information. So I don't know if anyone was in the other session this morning, but or maybe you noticed from your rooms, but the internet in the whole hotel went down for about half an hour. So hopefully that won't happen today. But there's the Wi-Fi information. We have a photographer, Cara Montes, who's here taking photos throughout the time. So anyone who has registered with a child, you should have received a waiver to sign. So we're going to use photographs from the conference in social media and other GRI material. If there's anybody who doesn't want us to use their photos that way, if you can ask at the registration desk for a green lanyard, and then when we see those photos, we just know not to use them at all. A couple research opportunities to mention here. So the first one is a blood draw. So this is something we're actually really fortunate to be part of this. So we're part of an organization called Combined Brain, which represents about 60 different neurodevelopmental disorders. And they have through Combined Brain, we have a consortium of industry. And every year, industry anonymously says which are the diseases they want to focus on. And in the past, the GRI disorders haven't been part of it. This year, at least 3 industry members identified GRI disorders as something that is a priority for them to do R&D on. And that means that we're eligible to participate in an industry-sponsored study called a proteomics study, where the key thing that they're doing is looking for biomarkers. And so the 5 genes, we need at least 20 samples for each gene to be able to participate. So, and they all have to be collected in the US. So the genes, we're focusing on all of the genes where we know we have enough patients in the US. So it's GRIN1, GRIN2A, GRIN2B, GRIA2, and GRIA3. The numbers for GRIA2 and 3 are smaller, so we're really having to push those families especially to participate. But everyone who has people with GRI here should have heard from us already to try to see if you'd be able to participate. But if you're, but it's still possible to sign up. If you're here, you can ask at the front desk, and there's a consent there, and we can get you signed up. But also, if your kids aren't here, we're helping people to get to Combined Brain collection sites, and we can also do a home collection at your house if you're in or near a city. So please, we encourage everybody to take part in that. Oh, the other thing just to mention that in addition to the blood samples from the patients, we're also looking for age-matched controls. So it doesn't have to be related, but if there's anybody here who has a child with them who does not have GRI but is under 25, or even any parents that are under 25, we would love to get a sample from you that can be matched to one of our patients to do that analysis. The other thing that we're doing is for anyone who can participate is a nasal swab. This is a much simpler way for a different kind of study, quick and painless. It's the same consent, so you still have to go through the consenting process. But that for both of these, you can go to the medical room between 8 and 4, or go to the registration desk or the GRI table to learn more. The medical room is when you're coming towards here from registration, if you turn left just before the first, just after the washrooms, I guess, down that hallway is where the medical room is. And then finally, there's research for GRIN2B families. So I sent emails to all of the GRIN2B families. There's one focus group that just happened. There's another one that's happening tomorrow between 12:30 and 1:20. And then on Saturday, There's a muffin tasting and app demo, so I'll just explain why there's a big happy blue muffin there. But the thing about this study is that it's looking at GI issues, so gastrointestinal issues. And one of the ways that they track these GI issues is by giving Grin2B kids these blue muffins, and then the parents watch for when it comes out, because it comes out a different color than the typical brown. So we have some blue muffins. If you want to try it yourself, you can come to that meeting on Saturday. We have our auction, our annual auction going on. There's a lot of really great items that are there. So just to highlight some, you might have seen our sponsor, Abrams Nation, that has this safety sleeper bed. There's one there that's up for auction. It's really a great product. I bought one the last time at the auction, so I know my kid does really well in it. There is, which family made?
My aunt.
Oh, Brittany's aunt. Okay, Brittany's aunt made the blanket in the Cure Green colors, and then there's a guitar signed by Britney Spears. Or no, not Britney Spears. It's signed by Taylor Swift. Okay, yeah. So I think she didn't actually. I know. I didn't know you want the Britney. You want the British. Okay, yeah. Taylor Swift. Yeah. Yeah. So that's. I think that one starts at $2,000. But yeah. So go check out the auction. Is it an easy link, Britt, or how do people find it?
Super easy link. It is.
Okay, when Brittany finds it, she'll tell us. Okay, so here's how we're going to start things off today. This is an Understanding GRI session, so we're really going to try to do the basics. And so we've got 3 speakers. The last of 2 are much more knowledgeable about all things scientific than I am. Ian Coombs has, you just told me what your PhD was in. Biochemistry, a PhD, and Jen Bain has both an MD and a PhD in neuroscience, but I'm going to try to, so they can fix all my mistakes, but I'm going to start by trying to tell you a little bit about genetics and an introduction to GRI genetics. So so here's how the time will sort of break down. So first, just in terms of some background, just some really basic science. So we all have trillions of cells in our body, and in each cell we have these chromosomes, and we have 46 chromosomes, and within the chromosomes are hundreds of genes. And so the genes that we're talking about today are some of those, and then the genes are what makes the proteins that are kind of the building blocks for all of us. Or they have the instructions for it. And so the process kind of works like this, that the gene gets pulled out of gets copied from the DNA into RNA. The RNA makes the ribosome, which creates goes to the ribosome, I guess, that creates the protein. And then in the case of our NMDA receptors, there's always 4 different proteins that come together to make an NMDA receptor, an AMPA receptor, a kainate receptor, or a delta receptor. And so there's always, in this case, there's always 2 GLUN1, the GRIN1 proteins, and then 2 other ones. And so this is what the chromosomes actually look like under a microscope. And so DNA has its own language, and it communicates. So just like when we're talking, we communicate using the letters of the alphabet. In music, there's musical notes. In DNA, it communicates actually using chemicals, and we kind of simplify that by referring to the chemicals with 4 different letters, A, C, G, and T. And it's kind of like, and so, sorry, for each of those chromosomes, or sorry, each of those 4 letters can spell words using just 3 letters. So every word in DNA is a 3-letter word using those letters. And so if you look at this chart on the bottom right, you can see The left column is the first of those three letters. The top column is the second of those three letters. And the letters along the right is the third. So for example, if you look at the G on the left and the G on the top, you can see that any three letters that start GG will always make a glycine. But if the second letter there changes to a C, that's good, or sorry, to a G. Wait, G. Yeah, the second letter changes to. Now, see, this is why you don't start with a non-expert. Anyway, okay, so I guess it's is it the first letter? If the first letter changes to a C, then instead it creates the arginine. And so that's what, in my son's variant, what's usually a glycine becomes a arginine, and then all of these amino acids come together to build a tower, but if there's something that's wrong, then it kind of can all fall apart. So it means that, like, even though we have all of these different letters in our genome, in the case of many patients, most patients with GRI disorders, it's just one of these letters that is an error, and it causes kind of this tower that's built to fall apart. And so the reason why I kind of think of it as like not just a tower of Lego, but it's kind of a complicated tower that's also held together. It's also held together by electrical impulses, electricity. And so some of these amino acids are positive and some are negative and some are big and some are small. So I put these like magnets in this image to kind of show that, you know, if you change one thing, what could happen is that just like if you took a magnet out of something that was built like this, the whole shape of it could all change. And that means that it could be like a major difference in the protein. And when it comes together into the receptors, it could also be a major difference. And so this is the most common diagram of an NMDA receptor. And if you look at one for an AMPA receptor, it looks similar. You can see just on the top chart there the number of bases and the number of amino acids. So a base or a nucleotide is each of those three letters, and then the number of amino acids is the word that's spelled by those three letters. So you can see that in the case of the GRIN genes, the protein made by, or that GRIN1 is a smaller gene than the other three. And so there's also different kinds of variants. And the most common kind that we see in GRIN disorders is de novo variants. And so that means that it's not actually inherited from a parent. What usually happens in the case of GRI disorders is that either right at when the sperm and egg come together, that's when one of those letters becomes mixed, or just after that, that's when it becomes mixed. We also have some of the other kinds that are not as frequent in GRIN disorders or GRI disorders. So autosomal recessive inheritance. So this is one that happens in GRIA3, a specific kind that is an X-linked recessive. So where the mother can be a carrier of the variant, but because she only has one copy of the X gene, it actually doesn't cause any problems in females, but then in males, or is it the opposite? It's the opposite. She has two copies, so she's okay that she has one good copy, but in the male, when it gets inherited, it can cause a problem. And then we also, again, even more rare, we have some autosomal dominant inherited genes where a parent can be a carrier and maybe have minor symptoms, and then the child, for reasons we are not entirely sure of, could have more significant symptoms related to the disorder. So first 6 takeaways, these are the points to remember that there's 4 letters in the genetic alphabet. Every word is 3 letters long, and each word tells the body to build an amino acid. Those amino acids are like Lego pieces with magnets that kind of come together and make the protein. And every GRI receptor has 4 proteins, and then most GRI receptors are de novo variants, so they're not actually inherited. Okay, so now on understanding your genetics report. So there's 3 ways people typically can get diagnosed with GRI disorders. The most common by far is an exome sequencing. So if you think of your whole DNA, the exome is just about 1% of that, and that's the part that actually has the instructions for making the proteins. And so most of the time you can identify a GRI disorder just by looking at that 1%. That's exome sequencing. Some people have had full genome sequencing where they look at the whole 100%. And there's a smaller number of people that have been diagnosed either on epilepsy or intellectual development panels. And so I'm going to skip some of this for time, but I'll just jump to this part here and just talk about the 3 most common types of GRI disorders that come out of this misspelling that we talked about. So if you think of this, the way that the genes are supposed to work, if someone has 2 healthy copies of a gene, it'll create 2 healthy copies of a protein, right? So that's what most of us have in most of our genes. And so that's good. So this is like I think of it in terms of sometimes like a volume switch, because the way that these receptors that the GRI genes code, they basically are responsible for communication between neurons. And so if the communication is at the right level, everything works. So the volume's set just right. But in the case, but there's also some variants where there's this one good gene, and then there's another gene that is broken, and it's broken in the way that it doesn't create a protein at all. So when this happens, it's something called haploinsufficiency, where just one copy of the protein gets made, and that can lead to problems. And the problems that happen with that are that the volume is turned down too low, and the communication just doesn't happen enough. What's even more common in GRI disorders is missense variants. And so this is one where there's, again, a good copy of the gene and a broken copy of the gene. The good copy will make one good protein, but then the bad copy, unlike haploinsufficiency, the bad protein still gets made, but it has a poor effect. And so in the case of what we call, you can hear the term loss of function, or dominant negative, that's another one where the volume gets turned down and the communication isn't happening enough. And in a gain of function mutation, it's like the volume is turned way up and there's just too much going through the channel. And again, there, even though it's like amplified, it's kind of distorted, and again, it results in problems. So when people talk about gain of function of loss of function, that's one way of thinking about how that works. This is my son's genetic report, my son Bryson. He's 18 now. So if you look at this, it says somewhere along here it'll say that it's a de novo variant. It'll say the two we can really pay attention to are the nucleotide and the amino acid. So we talked before about nucleotides and how those are each of those 4 letters that are the spelling. So it says at 1858, where there should be a G, there's an A instead. And then the next one, the amino acid, is saying that it says G620R. That means that at the 620th protein in GRIN1, where there should be a glycine, there's an arginine instead. And proteins are all different, or these amino acids are different sizes, and so an arginine is over twice as big as a glycine. The glycine is a neutral amino acid, where the arginine has a positive charge. And so, even though it's just this one letter change, it can have this effect where it can have a big impact on how the protein comes together and how it's shaped and how it functions. And then the other thing that is in genetics reports is it'll say at the right-hand side, it says it's damaging, probably. And so genetics reports will have one of these categories. Usually you'll only see it if it's pathogenic, likely pathogenic, or maybe a variant of uncertain significance. So if the last two, we all have like hundreds of these variants that actually don't cause any problems at all. So that they would be benign or likely benign. But pathogenic or likely pathogenic, that means that that spelling error or change that they've identified actually is causative. It is what's causing the disease. And so if they're really certain, it's considered pathogenic. If they're pretty certain, it's likely pathogenic. A variant of uncertain significance is one where there's not enough evidence yet to know. I was just talking with Dennis Lal, who's going to be speaking on a panel on Saturday, and he does a lot of work around this. And he said that roughly 20% of variants of uncertain significance eventually become designated as pathogenic or likely pathogenic. And so, again, just some more points to review, and then I'll pass it on. But so exome and genome sequencing are tests that are used to look at all of our genes at one time. While most of our DNA is the same between all people, we have many unique genetic variants, and most of them are benign. Most GRI disorders are caused by missense variants, but there are exceptions. And these ones didn't show up. But anyway, haploinsufficiency is when just one copy of a protein gets made. In missense, both copies get made, but if it's loss of function, then it's causing it to move too slowly. And in other cases, it's causing it to communicate or almost over-communicate, and it's gain of function. Oh, there. Okay. So that is me for now. I think what we're going to do is wait for questions until after all three speakers. Is that?
I just wanted to know if these presentations were going to be available so I can go over the stuff that you...
I can definitely make mine available. So all of these will be available through the conference website. And we can ask speakers if they'll share theirs, but that we have to leave to each speaker. Yeah. So Ian.
Ooh. I think it's safe. Okay. Do I have a laser pointer on this? Ooh. No, wasn't that one. Oh, sorry, I see. Yeah, it's the. Oh, wow, it's green. That's why. Okay. Hello, I'm Ian Coombs. I'm a scientific researcher at University College London. And what I wanted to talk to you about today was just some of the general ways in which we try and understand as much as we can about the variants that are found in your, I guess, generally your children. And I'm going to be talking about some little details about how we detect and how we analyze these variants. And there'll be a bit of overlap with what Keith has already done, but hopefully the idea is that at least there'll be certain bits which will firm up knowledge that you've already started to build, and there'll be other bits that will hopefully be new bits of information that you can take away. Okay, so I'm going to, Keith's already gone through a bit of the genetics. I'm going to be talking about how we can learn important details about each variant. And this comes from genetic information, so DNA sequencing. And then I'll also go on to talk about the technique that I use mostly in the lab, which is to try and work out the effect of each individual variant on the properties of the receptors, and this is done through electrophysiology. And then finally, in terms of making the models in which we look at these variants more advanced, how we can potentially get these variants into a neuron and actually work out what it actually does when it's at a synapse, which is where it's supposed to be. Okay, so first of all, DNA sequencing. Now, so how do we identify the variants? Well, first of all, of course, you need to look. So are the symptoms of the patient suggestive of a genetic disorder? And in all of our cases, obviously, it turned out to be yes, because you wouldn't be here if you hadn't had a genetic diagnosis. And so, as Keith mentioned, these can either be, 10 years or more ago, the majority of people would have been identified through gene panels. So that's where you look at the symptoms of the patient, and then you look at a focused selection of genes that are likely to produce a disease. And then you try and see if there are any mistakes that have appeared in any of those genes. Nowadays, the sequencing tends to be more extensive, and as Keith mentioned again, you can either look at the exome, which is where you sequence all the parts of the genome that actually get turned into protein, because only about 2% of the genome gets turned into protein. There's 98% is stuff that is just the bits in between that we don't care about so much because they're much less linked with genetic disorders. And then finally, genome sequencing, which is becoming more and more affordable now, where you look at absolutely everything. And if there are any changes at all of suspicion, you will almost certainly find them that way. Okay, and so I wanted to talk a little bit about the specific techniques that we can use to find out this information. So invented in 1977, there is a technique called Sanger sequencing, which you may have heard of. And this is still the gold standard, so this is still what we actually go to 100% confirm the genetic diagnosis that's suspected. And what the data looks like when it comes out, you have these little coloured peaks here, and they all correspond to the next base of DNA in the line. And so typically our bases are A, G, C, and T, and A's typically are green, G's are black, C's are blue, and T's are red, sorry. And what this does is when you have a particular stretch of the genome that you want to sequence in great detail, you can basically, with very, very high resolution and very, very accurately, you can take a specific stretch of the genome that you want to look at, and you can sequence it. And these typically give you about 800 or 1,000 bases of DNA that's read very, very accurately. And what it does is it amplifies a stretch of both copies of your gene equally. So in the GRI2 gene, what it will do is it will give you a read of this copy of the gene and this copy of the gene. And the result you get out at the end is a summation of these two things. So because both genes had an A in this position, you just see a pure A. And because it was just a G here, it was pure G. When it comes to the variant, you see you have a clash. You have two different readings coming from the same place. Okay? And this is very, very clear, very, very visual, and we can really be sure that this is definitely a true hit from our genetic screen. Okay, and so the next thing that gets used more and more now is a different technique. Rather than looking at a specific stretch that you're specifically interested in, it takes the entire genome, chops it to pieces, and then sequences all the little bits. And again, what you get is a summation of all the different bits of information. And by doing this, you can within a day you can sequence an entire genome, and this is more and more being used. The data you get is very brightly coloured and very, very pretty. But in a way, it's also still limited because you end up with just two different bits being summed together. And so if you have a variant, you effectively have this same thing, apart from you have it in a load more pieces, in effect. And the reason I'm telling you about these other ways of doing it is that now there's a new technique called nanopore sequencing. And this is a combination of DNA and electrophysiology, which is completely my thing. I absolutely love this technique. It's amazing. Rather than sequencing just a small bit of the gene, rather than fragmenting the gene into a million pieces and sequencing all the pieces, you can take a single unbroken strand of DNA, feed it through an ion channel, and rather than getting like 200 or 300 bases of DNA with this technique, up to 1,000 bases with this technique, you can sequence unbroken millions of bases of DNA by passing it through this ion channel. Okay. So the science of it is very cool. There will be Nobel Prizes. But why does this matter to us? Well, this particular form of sequencing called nanopore sequencing is opening up a brand new area of genetic medicine. And the reason for this is that, as Keith said, when you're making a protein, when you're making a glutamate receptor, you start with DNA, and then you make an RNA copy, and it's that RNA copy that then gets turned into protein. And for the majority of the GRI genes, except for boys with GRI3, you have two copies. And these two copies will always contain some minor differences. Obviously, the patients have got one very important difference that's in the coding region of their glutamate receptors, but there will always be lots of other minor ones as well that we've just picked up over time as a species. And so, and these are called single nucleotide polymorphisms, SNPs. They are individual parts of DNA that is different between all of us. And the vast majority of them do absolutely nothing, but they are in amongst the population. Now, these are commonly found in introns or non-protein coding regions and don't affect us. However, they still get converted from DNA RNA when we make protein. So we make a copy of this on our way to making the protein. And so why does all of this matter? Well, there is a particular form of medicine called antisense oligonucleotides where we can design a fake little bit of RNA that will bind to RNA and potentially break it down. Now, and they are selective. So even if you've just got one base different, you can design a drug that will knock down one copy of your RNA and not the other. Okay. And so in this particular case, what this means is that we have many, many potential places that we can potentially knock down the RNA. And this makes the existence of these SNPs means it's possible that we can design a drug that would knock down one copy of your gene, and we need to make sure that that is the bad copy. And so in this particular example I have here, we have ASO position 1, 2, 3, and 4. And so positions 1, 3, and 4 are SNPs. These are found in just the general population. You may have them, you may not. They're just randomly distributed. And there's also position 2, which is where that's the actual variant. And so for this particular patient, if they had this combination of SNPs, there would be a few different potential drugs that would knock down their bad gene. So ASO1A would knock down this copy, but not this copy. ASO2 would obviously target directly on the variant. ASO3A would knock down this copy and not this copy. And for this particular patient, ASO4 is of no use because you have two copies the same. But nonetheless, you have various different ways of potentially targeting it. And the reason I went through all that stuff about DNA is that because nanopore sequencing now allows us to have these enormous unbroken strands, it doesn't matter if the SNP in your affected gene is 20,000 bases off in one direction or 20,000 bases off in the other direction. Now we can identify which copy of the gene it is actually on. And we can work out with these ASOs, even if they're miles away from where the variant is, we can potentially knock down the variant form of the gene and leave the healthy form largely unaffected. And so I'm really, really optimistic that this approach will provide new treatments for GRI disorder patients going forward. Okay, so that is the genetics section. But now I want to talk about electrophysiology, which is what I do. And here is the brain. And when we think about glutamate receptors, we need to think on a slightly smaller scale than that. So if we zoom in, we can see brains are made up of loads and loads of brain cells which are connected up to each other. And when we zoom in a little bit more with the help of ChatGPT, we can see a synapse which probably looks exactly like this, but we don't know. And so what's happening at this synapse is that the first neuron, that the signals that are carried within neurons tend to be electrical. Where they meet each other at these synapses, you have a very brief chemical signal where a neurotransmitter is released from the first neuron onto the second neuron, and then that second neuron turns it back into an electrical signal to pass it on. And that's what glutamate receptors do. They catch the glutamate from the first neuron and turn it into an electrical signal. Yes, and this is incredibly important. They tend to be they are very short, and the whole process is really important crucial for learning and memory. Now, we can measure the currents that get produced by these glutamate receptors using electrophysiology. So if we think this was made by me, not by ChatGPT, hence why it's not quite so photogenic. Nonetheless, this is the very end bit of the first neuron. And then here is the very start bit of the second neuron. And so we have glutamate, the neurotransmitter, is held in these little packages called vesicles in the first cell. And when the signal comes in, this neurotransmitter is released to activate the receptors on the second cell, which I've got a mixture of NMDA and AMPA receptors here. Now, an important thing to think about with glutamate receptors is it's absolutely important that the signals they put out reflect the signals that have come in. And so if we have a situation where there's a weak activation, the first neuron has, it's not really fussed, it's got something to mention, hasn't really got anything to say, and we get a weak activation coming in, we might get limited glutamate release from the first neuron, which will only activate a few AMPA receptors on the second neuron, and you only get that small signal passed forward. If, on the other hand, the first cell has really got something it wants to say quite badly, then you end up with a much stronger activation. You end up with a larger glutamate release over a more prolonged period of time. And then you get activation of both AMPA receptors and NMDA receptors. Okay, and so that is the point of the glutamate receptors found there at the synapse. Okay, and so one question we want to know, and one question it's important to know to understand each case is what does each variant do to the electrical properties of the receptor? And okay, and so this was me last week filming my experiment. So here is the center of my electrophysiology rig, that's the microscope. On one side here we have a little electrode that comes in to make some electrical recordings, and another one, okay, that's just coming in from the left. Now this is mid-experiment, you may not have been able to tell, but this is through the microscope, and we have an electrode with a little bit of membrane with receptors on there being activated by jumps into glutamate. And just to the left, we have my computer. And this is the electrical current that's actively being recorded from that little system. And I send it from a situation where there's virtually no glutamate and there's no activity. I jump it into glutamate, the receptors activate, and then they turn themselves off very quickly as well. And this is the electricity that comes through one of these glutamate receptors. Now, the reason we try and examine this is because when you have a variant that's causing GRI disorder, typically it disrupts this kind of timing. So if we go back to our cartoon here, so weak activation gives a weak signal, strong activation gives a strong signal. And if we take a receptor that is overactive, so-called gain of function, then even when you have a weak little activation coming in, if you have overactive GRIA receptors, then you end up with a really big signal because the GRIA receptors have activated too much, they've recruited the NMDA receptors, the whole synapse goes crazy. And if you have gain-of-function GRIN, it's also possible that even a small activation by AMPA receptors would then lead to lots of signal coming through the GRIN receptors. If, on the other hand, you have loss of function, then even when you have a strong activation coming out of this first cell, you hardly, either the AMPA receptors are too weak or the NMDA receptors are too weak, and you're not able to pass on that signal. And so by understanding the properties of the individual receptors in each case of GRI disorder, we can try and appreciate which of these two situations is the case. And it's a really important step to identifying the best therapy, because if you have overactive receptors, you want to inhibit them. If you have underactive receptors, you want to boost them. And until you've done these kind of experiments, you don't confidently know which of those situations is the case. And so that's the kind of the bottom line of those experiments. Okay, I'm going to skip by this, but Dennis Lau will talk about computation and prediction and so on. Hopefully, the more we go forward, the less you'll need people like me to do electrophysiology, because you'll already have worked it out just from by looking at the gene sequence. Okay, and so I want to finish by just thinking about certain other ways in which we can look at these variants. So how am I doing for time, Keith? I feel like I'm going on. Yeah, I'm good. Okay. So we can take that little bit of membrane, like in the experiment I showed you, and that is just the receptor on its own, nothing else to confuse matters, and we're activating it and looking at how big an electrical current it produces. However, obviously, we don't exist as a bit of membrane on the end of a bit of glass. We have our brains are very, very, very complicated, and they're all interconnected, and it's a good idea to try and understand the variant in the context of an actual neuron and an actual complex of neurons. And there are several different systems. And I was tempted to put Disney throughout my talk, but I figured that we are probably closer. We're probably in the area of the densest population of copyright lawyers in the entire universe, I think. So I didn't want to go crazy. But very often our considerations will be either we can be poor old Donald Duck, or we could be Scrooge McDuck, depending on how we want to design our experiments. And in general, the expression systems that we work, they're kind of high throughput. We're asking a much simpler question. And you can kind of get the answer for hundreds of dollars per variant. If we want to look at neurons in a model system, like a rodent brain system, for example, this can cost between thousands and 10,000s. If we want to go to patient-derived cells, which I'll talk a little bit about as well, then that could be tens of thousands. And then ideally, right at the end here, we have a variant in a transgenic animal, and that is obviously a lot more expensive, but can tell us things that we can't find out any other way. And so in terms of this, what we want, we take our GRI disorder DNA and we stick it in a neuron. And we can either do this in neurons that are just sat in a dish, or we can do this in neurons that are in the context of their their tissue. And I really think I am going on, so I might kind of rush through these a bit. But the good thing about this technique is that we can use standard wild-type animal lines, which basically means these are well-established animal models that everybody has. And so you can just take a standard mouse and you can deliberately put your DNA in there and you can look at what happens. These cells tend to have mature synapses, so they're all grown up and they don't look weird. I'll come on to that in a sec. And we have a couple of different ways we can look at them, so we can pose our questions in different ways. The cons are the amount of gene expression is not controlled, so you can end up with 100 times more of your protein that you're trying to look at, and that can then lead to experimental errors. They're non-human cells, of course. And there's quite a lot of inconsistency in terms of exactly how much receptor you get in each of these cells. But nonetheless, at least you're looking at your channels in a neuron. Some of you may be aware of this principle, but it is possible to make what's called induced pluripotent stem cells. These are stem cells from patients, typically derived from skin cells. And what you do is you take them back to a very, very basic state where they can potentially become skin cells, or they could become liver cells, or they could become stomach cells, or they could become neurons. And when you do that, you can actually make neurons that are actually derived from actual patient cells, and so they are human cells with patient-specific background. This allows you for a better validation of gene therapy, because all the off-targets, the things you're trying to knock down, you know, but what you don't know is all the things you're not trying to knock down, and so this will help you work those out. And also you can even go just beyond neurons in a dish. You can even put different neurons together in different kind of ways that allow you to build a brain in a dish almost, or so they say. Alternatively, and on the downside of things, they're expensive and time-consuming. Synapses that you make commonly don't really look that much like synapses in the brain, and there are many different ways of doing it, and it's a very, very long process. And the most annoying thing about it is you need to give it an hour's care every single day for 28 days. And sometimes even scientists like to go to the beach or something, and you kind of, yes, your family aren't pleased. Okay, and then finally, the creme de la creme at the top of the pile is to make a model of a GRI disorder neuron. A mouse. I was going to use Nemo and Mickey, but I went with these. I couldn't quite bring myself to give Mickey a seizure. Anyway, and so we have a few mouse lines. Obviously, there's some labs working on fish, and by doing this gives you the best possible look at the variant in development. So the cells mature together in a more native environment. All the neurons express the variant at endogenous levels. You don't need to worry about overexpression or not enough expression. And probably most importantly, is it really valuable for preclinical stages of drug discovery and working out which variants are best to go into drug trials, and for example. However, of course, the downside is it's not guaranteed to match patient symptoms. In general, you will either have a mouse that's like completely unaffected or is dead and doesn't really look like the patient, unfortunately. Expensive to create and maintain, and often the patients are all heterozygous, but you might need to make 2 copies of the variant, and then it becomes a less good model. And so there are many, many different ways in which these things are imperfect, but nonetheless they all come together to allow us to find out more about GRI disorder. Okay, so just to summarize, knowledge is power. Basic science is necessary to identify and to advance new therapies in GRI disorder. The advances in genetic sequencing I talked about open the door to a new generation of gene therapy, which become more and more affordable, hopefully more and more available. Electrophysiology, as we do, allows us to diagnose whether variants produce too much or too little, which helps to choose therapies. And neuronal models allow us to investigate variants at synapses and are also very, very useful for helping us to develop drugs. Okay. And thanks very much to Kjorgren, who support the lab, and also the Medical Research Council. And thank you very much for your attention.
Hello. I'm much shorter than both of them. I'm Jennifer Bain. I'm a child neurologist. Thank you. I know it's a lot of words and talking right now. Actually, I think this worked out really nicely because you gave a little bit of genetic stuff, then you kind of talked a little bit more about what we're measuring and what the problem is. I'm going to go a little bit more on the clinical side. I'm a child neurologist in the office. I am not a basic scientist, but my goal is to try and interpret what the scientists are doing in the lab and bring that over to the patient side. I am appreciative of being here, and I don't know how to use this slide deck, but hopefully this will all kind of come together at the end, and then we'll have some time to do some questions. So what is a seizure? Anybody want to say? Yeah, what's a seizure? What do you think? It's a burst of electrical activity caused by a nervous cells. I know, isn't that great about activation? So what you were hearing about before was kind of balance, and so now we're just going to go a little bit more into that. As we talked earlier about if you took AI and it showed you all those little neurons, all those little cells in the brain, we have billions of these interactions or these connections in the brain. So that's each one of those little lights up there. And all this electrical activity, it can be either normal or abnormal, right? And so we use electrical activity to send signals from our brain down to our body to tell my foot to kick. But then also while we're sleeping, we have electrical synapses and these connections that help us learn new things and develop as we're younger. Unfortunately, when sometimes it goes a little haywire, you have a problem with that imbalance, and that's what we call a seizure. And so importantly, we're going to talk a little bit more about the chemicals, or the neurotransmitters is the big word that we use. And I know that was mentioned a little bit earlier today about the receptors. And so the receptors are kind of what the receivers of those neurotransmitters are. And so I have a really nice picture at the end that hopefully will bring it all together, hopefully. But the two main ones that we're going to talk about today are glutamate, which is the one that is excitatory and kind of activating, whereas that balances out with GABA, which is more inhibitory. And then we're not going to bring up AMPA because it's just going to confuse things a little bit. So in general, we're talking about balance, right? So we want to know that our input and our output are always kind of going equally. So if you have too much of one, which is your glutamate, where is this? Oopsies. Oh, man. Oh, there we go. So if we say glutamate is go, right, and GABA is stop, we just really need to figure out like the balance. Is the balance working? And if it's in balance, then you have inactivity, you have a problem, and you have too much overfiring and overshooting. And so a seizure is actually what happens is you're getting too much of that imbalance, right? So you're getting too much of that starting, too much of that go, that excitatory neurotransmitters or chemicals, and then you have not enough of that kind of braking mechanism, the inhibitory activity, that GABA. And so that electrophysiology that you were just hearing about from Dr. Coombs is actually measuring what is your in and what is your out, and how is that balance there. And when they sit there and they have the little, I don't remember what they call them, electrodes, and they can actually measure how much is going in and how much is going out, and is that balance correct? And that's really kind of the basic background of it. And seizures themselves are not really the disease, right? Seizures are just what we're seeing on the outside, where it's this abnormal balance of electrical activity. And it's really just a symptom of something else going on underneath. And so I want to kind of just make sure that we all understand, like, what is a seizure versus what is epilepsy? You've probably heard this before. Some seizures can be really hardly noticed, and some can be much more dramatic. And so depending on kind of where that abnormal balance of electrical activity is, sometimes you'll hear the term focal, right? So there's just one spot in the brain that seems to be, I don't know how to go backwards. One spot in the brain that seems to be really overactive, right? And then maybe that spreads to other areas. Whereas I think in most of the GRI disorders, or the GRI disorders, we're talking about a lot of the cells kind of all over the place have that genetic variant or that genetic typo that we heard about before. And it's causing those genes to be making abnormal proteins that are causing problems with those synapses or those connections, right? And so I think in general, we're talking mostly about these kind of generalized epilepsies. And then depending on kind of where you're seeing that abnormal electrical activity, you start to see different things, right? So we know that the right side of the brain controls the left side of the body. This area back here controls your vision, whereas this area up here really is more in control of your motor movements. So depending on where you're having that abnormal electrical activity, you might have different looking seizures. And so you might have a convulsive seizure, and if your loved one has a convulsive seizure, you might see something like a cry in a fall. You might not be able to interact with them. They're unconscious or have an altered consciousness. They might have shaking or stiffening of their arms or their legs. They might lose bowel or bladder control, and then they kind of slowly come back to it. Whereas there could be non-convulsive seizures, and these are also just as scary and just as problematic, where you might look like you're awake, but you're not necessarily fully engaged. You might be confused or wandering or screaming, or you might kind of lose tone. And so, again, we're not going to talk about specific seizure types, and actually you're going to hear more about that, I think, from Dr. Park later on this weekend. But understanding that there's different types of seizures and they happen in different parts of the brain is really important. So a seizure is just that symptom, right? But the likelihood to have a seizure, when you have more and more likelihood, that's when we're talking about epilepsy. So epilepsy means that you're at a higher risk to have seizures than somebody else who doesn't have epilepsy. And from our perspective, there's a group called the International League Against Epilepsy. Every couple of years, they decide like how they want to define epilepsy. And if you're ever interested in keeping up on that. But in general, it's typically somebody who has 2 clinical seizures, or more likely here is somebody who has a clinical seizure, but then they also have an EEG that's abnormal, one of those electroencephalograms, or a genetic disorder that we know puts you at risk for having epilepsy. And so we know in general, and we'll go back to it, that the genetic disorders are involving neurotransmitters that put you at risk to having seizures, and therefore you're at a higher likelihood of having an epilepsy diagnosis. You'll hear later this weekend from the Simons Foundation and the Simons Searchlight Consortium who's had seizures, what kind of seizures, what are the medicines we're using for the seizures. And so hopefully this is just an intro to get you understanding what are individuals in your group taking in terms of seizure control and seizure management. So again, a common cause of seizures is epilepsy, and epilepsy itself, also known as a seizure disorder, is the neurological disorder where you're at risk for having seizures that otherwise hasn't been provoked by other things. And we know this, but I like to always remind everybody that the neurological dysfunction in epilepsy really can kind of come at any time, and we're learning again more and more from the community. Thank you to Simon Searchlight for collecting this information. At what age are you at risk for having seizures? Just because I didn't have it when I'm younger, am I at risk for having this when I'm older as well? And we do know that obviously epilepsy is not contagious, it's not a mental illness, and it's not really indicative of a cognitive disability, but in the case of GRI disorders and other genetic disorders, there can be many co-occurring conditions and diagnoses. Just so everybody knows, there's more than just GRI and genetic disorders causing epilepsy. So some of the more common things that you can think about that cause epilepsy are prenatal problems, birth problems, in adults, often strokes, bad other things like tumors. So there are many other things that can cause epilepsy, but in this case, we're talking about just the way that the brain develops. And importantly, 1 in 10 people do have a seizure at one point over their life, not to scare everybody else in the room, but it actually is quite common. The question is really, what is the risk of epilepsy? And that's more in that kind of 1 to 2% of the population. And so really it's important, I think, for those who are here, many have either their loved ones have a seizure or at risk of having a seizure. We want you to feel empowered to know what to do if somebody did have a seizure. So knowing about seizure first aid, for those out there who have heard the term a seizure action plan, this is really important. Whether your child's had a seizure or whether they're at risk for having a seizure, that's something you can easily look up online. I don't know if the foundation has this, but you could easily get a link to the Epilepsy Foundation where they have one kind of pre-made for you, and you have that ready, just so everybody feels comfortable and confident if something did happen and kind of know what to do. You can have a seizure action plan even if you haven't actually had seizures yet, if you just kind of want to be prepared so you know what to do, so your caregivers feel comfortable. I'm not going to go too much into this, but in terms of kind of what happens if you do notice somebody has a seizure, right? We want to just stay calm first and foremost. We actually don't try to hold the person down. We just make sure that they're breathing well, make sure nothing is in their mouth, actually want them on their side or on their belly so that they don't have any vomiting, and that causes difficulty. Actually, don't do anything like CPR. We know that breathing is going to be irregular during the seizure, during that electrical activity, but they will breathe. And really, the most important thing is to get some emergency responders out there. So calling 911 and not offering them anything to eat or drink even when they're starting to wake up. In general, I think obviously if you have never had a seizure or seen a seizure, calling 911 is the first thing you're going to be able to do. But you're going to get to know who your loved one is and what your seizure action plan is going to be. Do I call after 5 minutes? Do I always go to the hospital? These are always questions you want to ask your provider so that you know what to do if that ever happens. But really, it's a matter of just making sure that person is safe. If they had this episode, what's the next plan? There are a lot of apps out there. These things might be helpful for you as a community clinically as well as potentially from a research basis. I know that EEGs, those electroencephalograms, a lot of people are getting them done all over the place, and there's some wonderful researchers in the field, like as you mentioned, Dr. Lal, who is collecting all these large data sets. How can we get all this information and kind of put it all together? But there are some mobile apps that are free, like Seizure Tracker, where you can kind of track these things and get more information, in addition to using things like the Simon Searchlight and giving all your information back. The more information we have about rare disease families, the better we can do giving back that information to you as families. All right, so now I'm just going to go basically into this, and thank you for sending these to me, Keith. Keith wanted me to kind of talk about, clinically speaking, what are maybe some of the medicines that people are taking that might be helpful. I think you'll see some of this in the Simon's data sets as well in terms of medications that are being used. So what I thought would be helpful is looking at these two slides. One is the 10 most helpful drugs or supplements, and so they're listed up here. And so I'll go through some of them up here, and then we'll go through the seizure medications, and then I'll finish up because I know that you guys have heard a lot over the last 45 minutes to an hour. So sleep, I'm not sure everybody's had bad sleep at one point or another. What we know is that the sleep is controlled by the brain. So it's not unsurprising that anything that involves the brain or dysfunctional brain disorders, whether that's epilepsy, autism, developmental delay, intellectual disability, puts you at risk for having problems with sleep. I'm a clinician half of the week, and I would say every single day I have at least 3 or 4 patients that talk about sleep and sleep problems, not only for that child, but the whole family. So we know this is definitely a quality of life issue, and it's certainly a challenge that should be discussed at every single visit, and it can be discussed at every visit. Problems could be anywhere from falling asleep to staying asleep to frequent waking up, as well as just general daytime sleepiness. So talking about this regularly is important. Melatonin, so these are the 3 medications that were listed as like sleep medicines that I think probably is most applicable here. So melatonin is a natural supplement. Your brain normally makes melatonin and it secretes it regularly throughout the day. And right before you go to bed at night, it peaks. And it says, time to go to bed. Does anybody know one thing that could actually suppress the natural melatonin? I'm trying to keep you guys engaged. Huh? Screens, who said it? Yay! So screen time will absolutely suppress your natural melatonin release, all right? So I don't know about you, but if I'm on the couch and I fall asleep and then I'm like, all right, now I'm gonna go upstairs and I sit down and I'm like, let me just scroll, just like for a sec. 25 minutes later, I look and I'm like, how did that happen? Because I was totally asleep like 20 minutes ago. So natural melatonin release is suppressed by screens. So if your kid, the first thing you should do is just turn off all screens at least an hour before bed for everybody, because that actually will help with just that basic stuff. And that's hard sometimes because maybe that is something that you use to kind of calm down at night. Unfortunately, the problem is that it kind of peaks when you're ready to go to bed and then it kind of teeters out. So if you've ever taken melatonin, it helps you fall asleep. It doesn't necessarily help you stay asleep. That being said, there are now more formulations that are called extended release, where the outside will dissolve really quickly. So it'll give you a burst at the beginning, and then it'll have a slow release formulation underneath it. And that used to be used more often in Europe, and now it recently has been coming over to the States. So that's definitely something to try. The problem is that it's a tablet, so you'd have to be able to, so I know you're like going like this. So when somebody can figure this out, please market it, and let's make some money for rare diseases for extended release liquid melatonin. It's gonna happen for sure soon enough. But that is something to think about. So it's definitely a safe one. People often ask me, is there concern about taking this long term? Will it suppress my hormones? hormones? Will it mess up my regular hypothalamic pituitary axis? The longest studies did look at specific hormones after a couple months of taking melatonin and actually did not dysregulate any of your natural hormones. In general, clinically speaking, I am not your physician, but what I do tell parents are it's absolutely safe to take it for 2 weeks, see if you can reset your cycle, and then try to back off for a little bit. And so using that, I personally have a bunch of different melatonin options for my 3 children sitting on the counter, and every once in a while they're like, I think I need a melatonin, and I'm like, that's fine. It was a tough day. Clonidine is another very commonly used safe medication that definitely has, I see some like fingers up for this one. It's a blood pressure medication, but it actually is probably used more for sleep as well as kind of anxiety, ADHD behaviors than anything else. It's quite a safe side effect, but it does make you sleepy, which is why we use it. It comes in a short-acting as a tablet and a long-acting. There's a newer long-acting liquid as well out there on the market. So I'm sure melatonin can do it because clonidine just did it recently. And in general, the only thing to know is that it is a blood pressure medicine. So if you're taking it regularly and all of a sudden you stop it, it can have a problem with your blood pressure. So make sure you're doing this. This is prescription only. Gabapentin is also another medication that is prescription only. This is actually a seizure medicine. You'll see it in a slide or two, but I'd say it's probably more often used for sleep, and it's very safe as well. It's also used for nerve pain, and so for a lot of people who have like neurological underlying diagnoses, gabapentin can be a nice medicine because it's very safe and it can kind of help regulate things. And then in terms of the other non-seizure med that seemed to be quite commonly used was Miralax. So pooping is a problem for a lot of neurodevelopmental disorders. There's definitely a lot of reasons why that's the case. Go to Eat Your Blue Muffin at a focus group because I think it's a really cool project to look at gut motility and see why do kids with neurodevelopmental disorders, including GRI, have problems with digestion. We know there's absolutely a connection between the brain and the gut. In addition to your brain, your gut actually has its own entire nervous system. And so when I was in med school a couple decades ago, there was like a chapter on that. Does anybody know the name of that nervous system? Anybody? The enteric nervous system. Have you heard that? Yeah. All right. So it used to be like a chapter in a book. Now it's like literally textbooks and literally, entire classes on this. I'm sure you can get a PhD on the enteric nervous system now. So I bet in 10 years we're going to even have more information. But Miralax actually doesn't do anything to the gut nervous system. What it does do is just try to pull as much fluid and water in there as possible. So it's definitely commonly used. It's definitely safe to use. And so for many people, I think most people have said it's helped at one point or another. All right, and then I wanted to get into top meds per category. So in the top left, we talked about the top meds actually going into those receptors themselves. We talked a little bit already about the Miralax and the other GI medications. I'm going to defer the neurostorms and a lot of these epilepsy meds for tomorrow, but just to kind of give you hopefully just like a foreshadowing for what you're going to hear tomorrow from Dr. Park and Dr. Kaufman, you'll hear more about these drugs. But I found this really great picture, which was not ChatGPT. It was actually published in a journal last year. So we're going to go back to what we heard about earlier, right? So you have these 2 nerve cells and you have them connecting. Does anybody remember what the connection is called? Synapse. Very good. All right. So here is a neuron. Here is the synapse. And here's the other neuron. And look what we're looking at down here, all of the receptors. And so NMDA and AMPA, those are the 2 receptors of the GRIN disorders. And what I like about this is that it shows where the mechanisms of action are for all the different seizure epilepsy medications. Again, if we talk about the inhibitory synapse, right, the stop, versus the excitatory go, we need to make sure this is balancing out really nicely. And so the signals come from one of your nerve cells, then you make all these little proteins, they go into the synapse, and then they come across to the other cell. And the goal of these medications, because you have too much of this activity and not the correct balance, is to really control this synapse at different locations. So again, these are your receptors that we're talking about in terms of what are the GRI disorders, what are the gating problems, and why are they either working too much, right, a gain of function, or not working enough, a loss of function. So in terms of top 10, I'm going to go in the order of what Keith gave me. So the top medication that seemed to be effective for GRI disorders was valproate, which specifically works in this inhibitory pathway on the GABA. The second was perampanel, which is Fycompa, which is working more on that AMPA receptor down there. This is called Keppra, and a newer version called Briviact, so levetiracetam or Briviact. This specifically is just looking on the receptors. It actually doesn't even care what neurotransmitter per se. It's just saying to all those neurotransmitters, we're not going to let the vesicles go, so it's a little more general. Up here, the other two that were listed are, I believe it was lacosamide and zonisamide, though I'm sure a fair number of people are also on Lamictal, which are going way up top, up to those sodium channels, so not necessarily related down here, but just more up at the top of the pathways. Many people are on benzodiazepines. The two that were listed here are clonazepam and clobazam, and what they do is they increase the GABA, which is that inhibitory one, so these can be really helpful. Benzodiazepines are also those emergency medications. Remember, we talked about the seizure action plan everybody should have. So often a benzodiazepine is used because it's really strong and it works really quickly. The downside is that it often can kind of turn everything off, including your breathing. So you need to make sure you're very careful about that and making recommendations with your doctors. And that was the other one that I put up there. Down here is topiramate, which also works on the GABA receptor, so a different pathway. And then more recently, there's individuals who's reported using cannabidiol, which basically means we don't really know exactly how this mechanism of action is working, but it certainly is being explored more in the genetic epilepsies. So that's actually it for me. I'm going to leave it open, but I thought this was a really nice, if people want to look, this is not my article, but I thought it was a really nicely done picture. Okay, thank you. Miss that.
So we'll move to Q&A, and anyone who's online can ask questions in the chat, and Brittany will read them out.
Hi there. I was wondering, one thing that I didn't know, we're, quite a ways into our journey, I think about 10 years at this point, but one thing I didn't know initially was to videotape when a seizure happened. Nobody really told me that. And so what I wonder is, on the seizure action plan, why isn't that one of the things that is, broadcasted everywhere for everybody to know? And then the other thing is the status epileptus. I'm not sure how to say that, but those are two things that I really didn't know in the beginning. So I just want to kind of ask about it because I also think it's good for everybody else to know because I didn't.
Thank you. So even before COVID, I had a saying. I said, use technology for good, not evil. And so that's beautiful. I think that's a great idea. I think seizure action plans are often used for schools and for caregivers, so I don't think everybody usually thinks to videotape it, but you're 100% right. And I'd say especially with the GRIs where you're having these neurostorms, these movement disorders, is that a seizure? Is it not a seizure? Is this epileptic or non-epileptic? Videotape everything. I mean, we're all paying $9.99 for your Apple every month, so you might as well utilize as much of that and then never do anything with it. But I think, yeah, if you can definitely use that regularly, it's really important. So status epilepticus is a nice long term for a seizure that is hard to control. In general, because parents always say, well, why can't I use the emergency medicine right away? Most brains, even brains with genetic disorders, can stop a seizure in a minute or two. right? We use the term epileptiform discharges or sparks. I like to use the analogy of like a match, like you strike the match, and when you have all those like mist circuits, you have like just little sparks, which is like the electrophysiology that's picked up. And if those sparks get big enough, that's when you could get the big forest fire, right? And those little tiny sparks, most brains are able to like get rid of it, right? And then the medicines you're on are supposed to be dampening out all those little sparks or discharges. When you get to about 3 to 5 minutes, it's really hard because you already have started the forest fire. And so most of the time when you get to 5 minutes, the studies show that it's harder to break that seizure. Some people say 5 minutes, and then there's other studies that are 15 minutes. And nobody's gonna do a study letting people seize 5 minutes or letting them seize 15 minutes. So in general, we typically aim for that 3 to 5 minute mark, where we say if your seizure hasn't stopped in 5 minutes or look like it's going to stop, you should give the emergency medication. And then status epilepticus means that, yeah, that forest fire is out of control, and it's very unlikely the brain is going to stop it on its own, and that at that point you need to have a medication that will stop it. It is a forest fire, and as once it kind of gets out of control, it can be really hard to break those seizures. And so we really want to prevent status epilepticus because they can certainly be very damaging. Going back to the beginning of those discharges, it's like a kindling effect, right? Like if you're just sitting there and it's like a smoldering little fire in your fireplace, any little poof is gonna cause you to have a seizure, which is why whenever you're sick, miss a dose of medication, stay up late the night before, have any other stressor, have a surgery, any of those other stressors kind of puts you at a higher risk to developing a seizure. I think those were both of them. I will say, I didn't talk about this, and you know, I'm not going to make a slide on it, but if anybody's, I think it's important to hear the term SUDEP as well. And there's a lot of emphasis in the field. So SUDEP is sudden unexplained death in an epileptic patient or a patient with epilepsy. It is a term that was coined probably a couple decades ago by neurologists, where individuals who have epilepsy, and actually not even if you've had epilepsy, but we know you're at risk for it, for example, with a genetic disorder, sometimes patients don't wake up. And even if we didn't see the seizure, we presume that they probably passed away from something seizure related. There's a lot of research in the field. I am certainly no expert on this, but there's lots of recommendations clinically that we don't aim to scare our patients, but we do want to empower you to know risks and what are the reasons why we treat seizures. For individuals who have epilepsy, who are at risk for having epilepsy, the reason we want to go on medicine is that one of the top reasons for having SUDEP, or one of the risk factors, is not taking medication when we know you're at risk for having seizures. And so again, my intention is not to scare anybody, but to just really bring that up because I think it's important for communities to know. I hope that's helpful.
Hi, my name is Brittany. I'm the carrier for my son's GRI disorder, and I was really interested, Dr. Coombs, when you spoke on the nanopore sequencing. Does that mean because he does not have a good copy of the gene that he wouldn't be like a candidate for that research?
Yeah, for that particular one, no. Unfortunately, GRI boys are the only gene within the GRI family that they only have one copy. Yes, so if you were to design an ASO that would knock down the copy, there would be no GRI at all, which is not good. So there are some proteins out there that you can actually get by pretty well without any of, and so it wouldn't automatically mean, you know, if it's X-linked that it wouldn't be possible. But for GRI, it's not unfortunately possible. Yeah.
Okay, thank you.
Okay. Oh, yeah, sorry, yeah. Yeah, for a male with GRI, yeah.
Hi, I'm Lana Wong. So I have a follow-up question to something Keith had said during his presentation, that in the genetic report, if something is identified as unknown, I think you said in 20% of those cases, it turns into something. So just confirming that is what you said, versus any of the other categories. Because now I'm thinking, in our genetic report, we definitely got the diagnosis for GRIN1, but we also got something for, if I recall, mitochondria. And since it didn't say anything, maybe it said unknown, I mean, that one I just ignored. Actually, the neurologist who gave us the results, we focused on the GRIN1 because it said likely pathogenic. But now I'm wondering if I should be paying attention to the other one and whether there could be any potential interaction between that and GRIN1.
Yeah, I'll give brief comments, then maybe Jen or Ian want to add to it. But yeah, so what I was told this morning is that, yeah, about 20% of those turn out to be pathogenic. I think that when a diagnosis is getting made, it's, there's what the sequencing report says, but then there's also what the symptoms are and whether they match what the report says. And so if if that one's pathogenic and the symptoms fit, then that likely is causal. It's possible that something else could also be impacting it, but and I think those numbers also vary, because I've heard that with like cancer genes, it's only about 10% that turn out to be pathogenic. So yeah, I don't know how solid that 20% rule really is.
Well,
So excited to answer.
oh, okay. Okay, don't worry, I won't spoil anything, I'm sure. I was also in the same conversation earlier with Keith. Yeah, we should qualify with Dennis exactly what he meant. I think he may have been saying that 20% of GRI variants that are currently classified as variants of VUS, he is able, just with the information he has, to automatically upgrade those to pathogenic. I think it probably, if you actually start to analyze all the VUSs in the database, more of them will become pathogenic. But just like without doing anything else, with all the knowledge he currently has, he can already send some of them to that next level. In terms of, yeah, I mean, the genome, we have 20,000 genes, and many of them, we will all have variants that we don't know the meaning of in lots of those genes. And so once you start looking at everything, you're going to find everything. And whether this other gene is, may have some influence on your child's symptoms, I mean, it's always a possibility. But yeah, hopefully the GRIN side of things is kind of very, very certain. But yeah.
So the way I think about it is time. With time, we learn, right? So back to just like thinking about genetic coding, right? When we started doing sequencing on lots of people, we looked at a group of people and said, okay, this is what we would consider this whole room. We're going to sequence all of us, and we're going to say we're all controls, we're all normal, we're all healthy, right? So then we just looked at everybody. Obviously, we're all a little bit different, so there's different changes, those SNPs, right? Like we all have little tiny minor changes. The question really is, what makes one of those changes a problem, right? Or like I say typo. So what typo is just different between us, or what is a typo that's problematic or is damaging? And so those terms, pathogenic, likely pathogenic, predicted in our algorithms to be damaging, right? Those are what we predict would cause a change in the protein, right? Because you changed your amino acid from this to that, and it went from having this charge to that charge, and it changed the whole protein and the way the whole thing worked. So number one, you have to look at what the gene is, right? If we didn't even know what that gene was, nobody had discovered that gene or described it 10 years ago, and now we know it. And then also the typo. Before, we didn't know anybody who had those typos. Now we found 100 people with those typos, and they're all healthy or they're all not healthy. only with the publishing of papers from Dr. Coombs and Dr. Lau and Dr. Chanellis are you going to learn what are the genes and what are those typos that really matter or not. And I think that's why it's really important to have a clinical team. Like some places you can go and see your clinical geneticist every 2 to 3 years and say, are there updates to my sequencing? Many people probably in this room even had a negative exome, and then maybe it was redone. The actual sequencing wasn't redone. The analysis and interpretation of that genetic code, because your ABCs are the same you're born with, right? For the most part, unless you have cancer, but like you're born with that code. The question is, do those typos matter? And the typos matter when science changes and when we update our science. So as new papers come out, as new publications come out, now you can, what they use the term reclassify and say, actually, yeah, that variant of uncertain significant, the VUS, actually is a problem because they published 5 papers in the last year. So the day before the paper comes out, your doctor doesn't know about that disorder, right? Your child had GRIN1 all along, but the doctors didn't know because the disorder was never even described, right? So that's why it's so important to be involved in communities like this where the researchers are being brought together by families to say, keep moving forward. Are there other variants that could be playing a role? How is science changing? How are we learning more?
Is there anything online, Brett, or?
I'm not seeing anything online right now. I will sneak in the auction, Galabid CureGrin 2025. We are auctioning off a seizure monitoring watch, if anybody's interested.
Hi, my name is Ashley, and this question is for Dr. Bain. I am interested in knowing, like, if you have 2 children that both have generalized epilepsy, but one child has a seizure, grand mal seizure that lasts 2 minutes and has to take emergency medication, or 5 minutes and has to take emergency medication every time. And then there's another child that has a grand mal seizure, but they only last 20 seconds. What is it? Like, where's the difference? Like, what's happening that causes one child to have such a severe, long seizure as compared to the child that it's the same type of seizure, but they're shorter?
Yeah, you have a great question. And I think, you could even step back and say, if you have in the Simon's database 20 people with the exact same typo and the same gene, why are they all different? Like, nobody understands. We call that heterogeneity, phenotypic heterogeneity. And I think that's where these large data sets are coming out, hopefully, to say, are there other variants that might be kind of playing a role? Or does it matter if it's a boy or a girl? Or does it matter were they born premature? There's probably, there's definitely different effects, and environmental or genetic, that are modifying why you can have the same people, like the same exact genetic typo, and such different people. Even within genes, right, you can have so many different changes, and that's why all this research is so critical to say, it doesn't matter where the typo is, what the typo is, when the typo happened. I don't know if you want to comment.
I have nothing to add. Unfortunately, I'm sorry. I thought something was going to come, and I went blank. I apologize.
Hi, my name is Jillian. I had a question for Dr. Bain. You mentioned in your presentation that epilepsy can occur at any age. We were told by our neurologist that if you haven't had epilepsy by age 6, and then the onset of puberty was another risk factor, but if you managed to get past those 2, chances of epilepsy were slim to none, except, of course, you get in an accident or something like that.
So it's a great question. So as a clinician, when I see a patient, I say, why do they have epilepsy? So if somebody came to me and they said, my child has autism, we know that they're at a higher risk for having epilepsy, when is that going to happen? And I'm going to say, well, in most of the studies of patients with autism, they have it when they're younger children or when they're teens, and those are the 2 blips, and then that's it usually. When we add the genetic component. Now we can actually start saying, okay, well, what about people with GRIs? It actually depends which GRI disorder you have. So it's not as simple as just GRI. And then it really does depend on the variant that you have, meaning is it a gain of function or a loss of function if you're at risk for having seizures. And then it's a matter of getting more and more data from these registries at Colorado and Dr. Lemke and Simon Searchlight to looking at 100 people and saying, when are they having their seizures? Hopefully we see trends. That being said, you can have the same person or two people with the same gene and you might still have a slight difference. So we can make predictions. But it all comes back down to like what's published in the literature, right? As a clinician, is your clinician looking at where are they getting their information from and making these recommendations? Unfortunately, any genetic neurodevelopmental disorder, you're always going to be at a slightly higher risk for your whole life, but there probably are peaks that are more vulnerable than others.